Welcome to our webinar series. Brilliant Minds is a webinar series sponsored by the Israel Cancer Research Fund, in which we share with you cutting-edge research being conducted by Israeli scientists whose work is sponsored by the Israel Cancer Research Fund. I'm Dr. Mark Israel, the National Executive Director of the Israel Cancer Research Fund. ICRF is a 45-year-old nonprofit that raises financial support for cancer research in Israel. And since 1975, over $72 million of cancer research in Israel has been supported through over 2,500 grants to Israeli cancer scientists at all the academic biomedical research centers in Israel. The title of today's webinar is Listen to Your Gut, Cancer, Immunity, and Gut Health. Of course, cancer of the large intestine, of the gut, is a major public health problem, and great efforts to prevent colon cancer are manifest in the pursuit of adults to have colonoscopies on a regular, if not infrequent, basis. And of course, there are other fairly common disorders of the gut that are marked by significant and debilitating symptoms. In the last several years, we have come to realize that the gut, which is in contact with the outside world, the world outside of the body, at both its beginning and end, is filled with bacteria and other microbes that have a role not only in the proper functioning of the gut, but also in mediating our immune system and our immune system's response to disease. Today, Dr. Geva-Zatorsky will provide some insights into the biology of the gut in her research, which is focused on how microbial contents of the gut communicate with and influence our immune systems. Dr. Naama Geva-Zatorsky is a principal investigator of biology at the Technion in Haifa. Dr. Geva-Zatorsky received her Bachelor of Science from Tel Aviv University with a double major in chemistry and biology when she graduated summa cum laude, and her master's and PhD from the Weizmann Institute, studying systems biology, which she completed with honors. And recently she concluded postdoctoral studies at Harvard Medical School. Dr. Zatorsky will be interviewed by Dr. Nancy Maizels, chairperson of Israel Cancer Research Fund's International Scientific Council. Dr. Maizels is a professor of immunology and biochemistry at the University of Washington in Seattle and has been the director of the molecular medicine program there. Nancy, I'll turn it over to you.
Thank you very much, Mark. Good morning, good afternoon, and good evening to everybody. It's my pleasure to welcome Naama Geva-Zatorsky as our speaker today. Naama's work has always been at an interface, chemistry and biology as an undergraduate, and then systems biology ever since then. She's received lots of national and international recognition. She had a UNESCO L'Oreal Award and Human Frontiers and EMBO fellowships as a postdoc. She's received the John F. Kennedy, Teva, and Barinholtz Awards for academic excellence. And at the Technion, she's been honored by the Horev and Alon Awards. What I find especially beguiling about Naama's research is that she's exploring the bond between us and the microbes that inhabit our gut. And these microbes are of several different kinds. The tiniest are little viruses that infect bacteria, and next up are the bacteria that infect us. It's easy for us to forget there are 10 times as many bacterial cells in one of us as there are human cells, and how we all live together determines our health or our disease. So I'm really delighted to have Naama here this morning. And my first question to her will be, Naama, how did you start doing this work?
Hello. I started actually with biology because I was always fascinated by nature. As a child, I lived in a natural environment in a moshav in Israel. Most of my time I walked around in the fields, picked up snails, ants, asked my parents many questions, and they also encouraged me to be excited about nature. So for my first degree, actually, that was the reason to study biology and chemistry, just followed my heart, my passion. it was difficult for me to study a profession. And since then, I'm there. Actually, this is the same reason I moved to the microbiota field, to the field of study that I'm studying right now, and also during my postdoc at Harvard Medical School, which is these bacteria that you mentioned that live around us. And I was always passionate about them because they're unicellular. We'll talk about them in a moment. They're unicellular, but still they complement many of our physiological functions, and I find it fascinating.
So start by telling us a little bit about the project that the ICRF has funded that's going on in your lab.
Sure, yeah. I was very fortunate to receive the ICRF Accelerator Fund, which is a grant for a bit, I guess, more, it's called Innovative Ideas. It's, well, I can say about myself more on the borderline of crazy. So that was about these bacteriophages, which are the viruses that you mentioned that infect bacteria. And how can they improve our cancer state? So we'll talk about it more later, but we see in the recent years that these microbes that live in our body complement many of our functions, and we study mainly the way they complement our immune system. And this ICRF fund was to study whether these viruses of bacteria, these bacteriophages, can fine-tune the way these microbes communicate with us. So can they be the drivers of the functions of the microbes? Because so we did have quite promising results when we submitted it, and more promising results now with this fund. And it's very exciting because it's really can be related to cancer, as on one hand, they can create an environment which is very suppressive that would not allow cancer to initiate and progress. And on the other hand, they can create an environment that is more stimulating the immune system and helping us fight cancer, since our immune system can fight cancer, but we need to move it towards this direction. So we really study this balance and how can we tune it towards either preventing the cancer, or once cancer is there, how can we improve the way the body fights it.
Tell us more generally about the projects that are going on in your lab and how this works.
Sure. I will share my screen. Yeah. Okay. So thank you. Thanks a lot also for this opportunity. So we're studying the bacteria throughout our body, and mostly in our gut. And how can we be friends with them? We believe they are our best friends. So microbes are living all around us. As Nancy mentioned, about a 10-to-1 ratio of microbes to human cells. They live throughout our body, on our skin, mouth, nose, ears, lungs. But the major location is the gut. And we see that they can complement many of our physiological functions, and they are important in many types of diseases. Some of them perhaps more reasonable, which is, for example, inflammatory bowel disease, because it's an inflammation in the gut where they are located, but also autoimmune diseases where our immune system attacks us. So these, our gut microbes can fine-tune this disease state as well, diabetes, and also cancer. In cancer, for example, very promising studies show that patients with specific microbes can respond much better to cancer therapy, and others respond less well. In this direction, there are several clinical trials that implant microbes from patients that respond very well to cancer therapy into patients that did not respond well, and those become responsive. So we see that really the gut microbes have a lot of potential also in cancer therapy. I think it's one of the most exciting fields in this relation between our gut microbes and our physiology and health state. So I mentioned that they are also inhabit our skin. In our skin, this mosquito is here because we see that the diversity of the microbes on our skin can affect our susceptibility to mosquito bites. And there are now creams with microbial products to repel the mosquitoes. The cute squid is a squid from Hawaii that Margaret McFall-Nye and Bonnie Basler are studying. Margaret McFall-Nye in Hawaii has shown that this squid has actually an organ for microbes. It's called the light organ. And it's because this organ allows this octopus, the squid, to be unseen, actually. So I think it's really a fascinating story. So although it's not exactly on the flow, I would like to stop for 2 minutes to talk about it, or even less. This symbiotic relationship between the microbes and the squid are really fascinating because the squid can actually go out at night. It will not have a shadow by the stars and the moon because these microbes shed light, they luminescent, and they cancel its shadow. So I find this relationship very fascinating, and it's one of the reasons that I moved to the field of microbiota host interactions. Bonnie Basler actually showed these molecules that cause the microbes to light up. And it's actually these microbes communicate between each other, they count each other, they shed this light, and then they cancel the shadow of the squid. So we also believe that like the light organ of the squid, we have a forgotten organ in our gut because we see that our gut is inhabiting many microbes, thousands of types of microbes, and they complement so many functions that we call it the forgotten organ. These elephants eat their own feces, and it's actually very common between animals. Also bugs. Some bugs know the purpose of life only after they eat their own poop. And it's actually very smart. And the study that I mentioned earlier about cancer immunotherapy is really this. It's fecal transplantation. It's at this stage, it's actually transplanting feces from responsive patients to cancer therapy. One of the indications for fecal transplantation is Clostridium difficile infection, which is an infection that causes a lot of diarrhea, like tens of times a day. And fecal transplantation relieves the patient at once. So after 2 days of treatment, there is no recurrence of the disease. So this is one example and the only indication that came out very fast to the medical field. But right now there are over 300 clinical trials on fecal transplantation and many indications, including cancer and also several types of cancer, not only colon cancer. The example that I gave was on melanoma. So how do we encounter these microbes? We first encounter them at birth, and we really see differences in babies that are born in C-section versus babies that are born in the natural way, in the vaginal tract. As fetuses, we are sterile, and when we are born, we start collecting these microbes from our parents, from our siblings, from our family, and so on. It's important not to be too clean, although nowadays with the corona, we do need to make sure we balance our hygiene. We see that our immune system is educated by these microbes, and its development is triggered by these microbes that we start to collect. We study this in lab, especially in relation to the immune system. And we work a lot with germ-free mice, which means mice without any microbes. Because they don't have any microbes, we can isolate them and isolate the mice and colonize them with whichever bacteria we are interested in. And then we can study how this bacteria affects the mouse, mainly the mice immune system. So we find that specific microbes tune up the immune system in different ways. And we studied this both in health conditions and in cancer, both as a prevention method and as a therapeutic method, because both the live microbes themselves and also the products, their molecules can become future therapeutics. This is how they look. So we developed a method to fluorescently label them. And this is actually inside a live mouse, inside the intestine. The green dots here are the live microbes. So this method allowed for the first time to visualize live microbes inside live mice. The blue here are the crypts and villi in the gut. So this is how the gut looks from the inside. And the green are the microbes, and that's where also the food and the feces pass. And thanks to this technology, we can study how the microbes communicate with the immune cells of the mouse. We also add bacteriophages, the viruses of the microbes, to this system. So this is how they look like, these phages. And these phages can pop the microbe. They can actually infect the microbe and cause death of the microbe. And the ICRF grant that I mentioned at the beginning is exactly on this, because thanks to this popping, some of the microbial molecules are more exposed, and that changes the way the immune system is activated. So this is actually a tool that we use in order to fine-tune the way we activate or suppress the immune system. If we suppress it, we can prevent the initiation of cancer. And if we have cancer, we can tune it up in order to help the immune system to fight the cancer. I think this is the last slide on this topic, just showing the potential that we believe that in the future, pharmacies will be full of microbes, phages, or microbial products that can be used to tune our immune system and help our health.
So is there anything we should be taking now, Naama?
It's a great question. There is now a little bit of a conflict because there are probiotics on the shelf, but I'm from the side that says that we don't know enough about the microbes in order to say which probiotics we should take as healthy or as depends what is the health state. I think very soon we will have more fine-tuned and probably also personalized probiotics depending on our health state. So I think there is a lot of potential here, but unfortunately I don't have an answer of we should all take this probiotics today.
So one of the things I've been struck by in Israeli science in general, and certainly your lab, is that many people feel very flexible and they know that they can use their intelligence and skills and creativity to solve lots of different kinds of problems. And of course, we're all thinking now about Corona and how we can address that. And your lab has had quite a hit in the last few weeks by developing a simple home test kit for coronavirus infection. And I know everybody would love to hear more about that if you don't mind.
Sure, thank you. And thanks for your very kind words. So actually this is, it was an off the track project. When Corona started, all my labs sat and thought a lot, how can we help, and this is what we came up with, which is to apply a method that includes reagents mostly sold by NEB in New England, to detect the coronavirus directly from patients with no professional experience required, with very low cost, under $2, maximum under $5, and a very quick and easy test. So this test includes a saliva test. It can be self-tested, so you can test your own saliva. That's why it doesn't require any safety, because you anyway touch your saliva most of the time. So, and then very short incubation step at room temperature, then very simple reaction. In one, we actually did it from, in Israel, it's called tummy bar, the bars, the water bars that are everywhere. You can press hot and cold. So we just added hot water and then a little bit of cold water. And the result is actually seen with your eye. So pink becomes yellow if you are positive. This is how it looks like. When it's negative, these are real samples. It's pink. And when you're positive, it's yellow. The result comes up in about 40 minutes. So it's not very, very fast, but it's fast enough. I imagine it as waking up, spitting to a tube, performing one very simple step. And then once you're ready to go out to wherever you are going that day, you already know if you're positive or negative. It was a very adventurous experience, because once we realized it, actually friends of mine were telling me, have you thought about this? And then it took me a couple of days to think about that. And then we talked in the lab and thought how we can turn it into an actual protocol that works directly on samples without any purification steps that we usually perform in the lab. And then a team of about 10 people in total, some from home, some that came physically, worked like night and day. We really didn't go back home, slept on the couch with really like 5 hours and 5 days of sleep. It was really crazy, but very fast we came up with these results and published it so that the world can continue and make it happen. And I think it has a lot of potential.
Do you think this will be made commercially available so that we'll all be able to use it?
Yes, I think so. There are many that are showing interest in it. As is, it's possible as well. As you can see, it does require a couple of steps, not very, very easy, but still, I think it will be better if it will be a device that we will only spit, close it, and then get back to it in 40 minutes. So from around the world, people are actually contacting us both with ideas to apply it on a device and to help us get an FDA approval and get it to the whole world, Africa, United States, South America, many Greece, many contacting us. So I hope, yes, I think it's possible, and I hope it will indeed help.
I think we're all very, very excited about this possibility. That's really wonderful. Yeah.
Thank you very much.
Yeah. So now what I'd like to do is to open the floor to questions, but I have one that just popped up on the chat that I'll go ahead and ask before I turn it over to Mark. So the question is the obvious one. What's the time frame for making this test available?
It's a good question. It's a difficult answer because we are dependent on FDA approvals, for example, although it seems like it's really possible. So I hope weeks. I mean, yeah, I hope weeks.
And are the reagents things that one could just go out and buy? If I read your paper or someone else read your paper, could we order the components?
It's possible, but I need to be very careful because, I need to be very careful because if you are, I can say that if you're positive, then you're positive. We didn't have any, we have very few, only one false positive in all the 200 samples that we tested. Most samples that we tested were from swabs because that's how people test right now in the hospital. So these are the samples we had. We did get approval for a few saliva tests, and those were also working very well. So if you're negative, if you're negative and you feel bad, I wouldn't say you're for sure negative. So I think it's better to test once we get the FDA approval.
Sure. Yeah, one of the questions that just popped up on the chat was, what's the false negative, right? And obviously you need bigger numbers and more different people in different places to get the best kind of data. But this is just tremendous. Yeah.
Thank you.
Thank you for sharing that with all of us. Okay, so Mark has been collecting questions. I'll turn it over to Mark now. Thank you, Naama. And we'll hear what the audience of over 140 people have been thinking about. Mark?
Great.
There are numerous questions, so I think we'll only have time for a few, but one of them asks about what other bodily functions microbes might affect and what other kinds of diseases. So beyond the gut and so forth, is it thought that there's an effect on neurologic disease like Parkinson's disease or any other major bodily systems?
Yes, thank you for this question. Yes, the gut microbes can affect many physiological diseases, Parkinson as well. There were studies showing their effect on autism, for example, and Alzheimer. This is less my field at this point, so I would not elaborate much on it. But yes, they have been shown to have effects also in the gut. It's called the gut-brain axis, and I think it's promising as well, very much.
Thank you. Now, a very significant public health problem that's pointed out by one of our listeners in Canada, actually a board member, is antibiotic-resistant bacteria, especially these acquired infections in hospital. And the question is there reason to believe, or is it likely that bacteriophages could be used as a therapeutic modality to actually act in an antibiotic manner to kill bacterial infections that we currently don't have antibiotics for?
Yeah, thank you. It's a great question. And actually we have a publication that is coming up in, it's already accepted and will be come up in clinical infectious disease on exactly this. So on patients that acquired antibiotic resistant bacteria that are resistant to antibiotics, we performed a clinical trial and added, so to the patients, we added fecal transplantation. We gave them feces, fecal transplantation, and found that the fecal transplantation can eradicate the antibiotic resistant bacteria. In the fecal transplantation, I should say, like Nancy explained at the beginning, the feces is composed of both bacteria and viruses. So here we see that it helps. We can identify the microbes that were enriched in the patients where the microbes, where the antibiotic resistant bacteria were eradicated. So bottom line, the fecal transplantation helped to eradicate the antibiotic resistant bacteria. And now we're performing experiments in mice where we actually add phages, bacteriophages, the bacterial viruses, specifically against the antibiotic resistant bacteria to the mice, plus or minus the whole feces transplantation. And, so we see that in this system, the phages alone were not enough, but the combination of the phages with the fecal transplantation was better, at least as of today, compared to the fecal transplantation. So there was something in the phages that allowed the eradication much better. I mean, the phages probably killed the antibiotic resistant bacteria, and the fecal transplantation helped replenish the microbiome. Yeah, a long answer.
Yeah, but very interesting. In this regard, there are quite a number of questions about the therapeutic potential of bacteriophage beyond what you just articulated. Is it imagined, is one of the strategies to put in a capsule bacteriophage, viruses that infect bacteria, and have people swallow it and infect their own guts with bacteriophage? And if so, is there any concern that this might be unsafe and that there are both safe and unsafe kinds of bacteriophages?
Yes, it's a great question. Phages, it's possible, and there were several studies in the past 5 years or even less. It is an emerging, I mean, bacteriophage therapy is not extremely new. It was there for tens of years, and in some places of the world, they use it a lot, mainly in East Europe. It's very, very common to use phages as phage therapy to eradicate bacteria. But in the past 5 years, it's emerging throughout the world. One thing that I would like to say is that the phages can modify themselves very fast. So if we add phage to bacteria, they can, both sides can develop resistant to each other. So the bacteria can be susceptible to the phage kills the bacteria, but after several days, some bacteria will become resistant to the phage, then the phage changes and becomes again infective to the bacteria. So in some of the systems, we see oscillations, like phage infecting, bacteria die, then bacteria become resistant, less phages, then phage become infective again, and so on. So it's probably not as simple as adding a phage or even a phage cocktail, but there was a really nice case study in San Diego where they actually did like evolution in the tube. So they gave the patients phage to eradicate the bacteria. They then raised new phages against the bacteria that became resistant until they really eradicated the population of the bacteria. So not simple in most cases, but not impossible. It's possible. These days, I would say it requires a little bit of tweaking and work every like close to the patient, but it's possible. There were also cases where one treatment helped. In Israel, there was a case with a patient that had to get his leg amputated, but didn't agree. So instead of, so they gave him phage therapy, and actually it helps. It helped. After 3 days, he walked. So a lot of potential.
Mm-hmm. Well, bacteriophage, of course, the whole concept of viruses that infect bacteria is not well known to the lay population. And I have many questions about that. I'm going to combine a couple of those questions into one thematic item to share with you. And I'm wondering, are healthy people, all of whom have many, many kinds of bacteria in their gut, also harboring gut bacteria which are infected by bacteriophage? And do these healthy people have a variety of populations of bacteriophages, of these viruses in their gut that are helping to balance the gut microbiome, the entire flora of the gut, and play a normal physiologic role?
Yeah, Yes, there are bacteriophages in our intestines, also, both in healthy conditions and in diseased conditions. One study showed that when the gut is inflamed, there are many more lytic bacteriophages, which means phages that really infect the bacteria. Some phages that we have in our intestines are dormant. They are there, but they're not infective, and some are there with their infectivity capability. It's still a new field in the microbiome, so these questions are right now being answered. We also look into how the phages affect our body, even regardless of the bacteria, and we see something there as well, which is quite surprising. But yes, the answer is yes, there are bacteriophages in our healthy intestine. They probably are there to balance the populations, and they probably have additional effects, whether actively or like these are cascades of effects, like they infect the bacteria, and that affects how our immune system is affected by the whole bacteria and phages.
Now, Naama, I know, and I hope our audience realizes, and I thought you were very cautious in your remarks not to sort of represent suggestions about health and what people ought to be doing. That probably is best left to a gastroenterologist. But there's a lot of interest and many questions about prebiotics, probiotics, and what people can do just in terms of their diet to improve their gut health. And so I don't know if you feel comfortable answering that question, but I think speaking to the role of roughage and fiber and so forth might be of interest and help to people.
Yes, yes. It's also a field of study. Of course, it's difficult to give everyone a recommendation, but in principle, so yes, prebiotics it's called the food of the bacteria. I guess everyone is aware of, and probiotics, the bacteria themselves. So yes, as you mentioned, the fiber-rich diet. I always like to say a diet with all the colors, all the natural colors is good. So the vegetables with all the colors and combination of proteins and a lot of fiber is, of course, that's good for sure, because it helps the diversity. We see sometimes that in some cases, it doesn't really matter if you have this or that bacteria, but it matters to have a diversity. So a high diversity of bacteria will make you healthier, and a high diversity of bacteria is fed by high diversity, high fiber food. I hope this helps.
Well, I think addressing the issue that eating a varied diet and having a variety of microbes is generally associated with a healthy gut. But there is a very interesting question here about whether or not the microbiome, whether or not the collection of bacteria in a healthy gut has really been defined. In other words, we know that you can collect a gut specimen and you can do DNA sequencing and you can make a long list of the different kinds of bacteria that are there. Beyond diversity, is there such a thing as a healthy list and an unhealthy list? And if so, is there reason to think that eating a varied diet and various kinds of probiotics might convert an unhealthy gut to a healthy gut?
So the last part was, is there a reason to think that high fiber will convert an unhealthy gut to a healthy gut? Yes?
Yes, and is a healthy gut and an unhealthy gut defined in terms of the specific kinds of bacteria?
Yes. So diversity is one, which is always very important. And there will be differences. So tribes and in South America that are lately studied on their about their microbiome diversity have high diversity, but very different microbes than the Western world, and they are very healthy. So I wouldn't say there was a list of the healthy Homo sapiens, no matter where you are, but I would say that there are some bacteria that if they come up in the sequencing of your intestinal content, it can say that they can lead the investigators or like the researchers or the doctors to understand if you are healthy or not. So some microbes that we see in the gut are definitely showing, giving us a red light, a red sign that perhaps there is a disease in the patient. Diversity, and yeah, there are, but it's not something that I can right now say because it also depends on the relationships between them. Like back to the beginning, everyone, right. Yeah, okay.
Naama, we're coming to the end, and I think I'll just, there are many, many questions about your very exciting new test and so forth, but one that sort of captures a variety of the questions is how sensitive the test is, how important is sort of viral load, the amount of virus. The fact that it was already examined many patient specimens, I think, speaks to the fact it's sensitive enough to detect what is possibly present. But have you made an attempt to actually define the sensitivity?
Yes, we did in the swabs samples. So yeah, it's a great question. The viral loads matters. So in very low viral loads, so the sensitivity at this point is not as good as the test in the hospital. In very low viral loads that can be detected in the hospital, this test will not detect them. But in medium to high viral loads, it detects. It can say to whoever, if there is someone in the audience that is aware of the numbers in the hospital. So in cycle 29 of the qPCR, which is the result in the hospital, and down, meaning from viral load of cycle 29 and lower cycles, which means higher viral load, this test can detect in over 97% accuracy. Again, in the negatives, there are very few, very low false positive, very low tests that shows that you're positive, but you're not. So most probably, and like 98%, if you get a positive result in this test, you're positive. If you get a negative result and you feel bad, I would suggest, you feel sick, I would suggest to test again. So the advantage is that this test can be like a surveillance because it will be very easy and very cheap. And we can test ourselves every other day or whenever we feel a bit insecure about our health state in relation to COVID-19. And if we're positive, it will indicate that we're positive, or it will probably in the future be another indication to do the actual test in the hospital. So medium to high viral load. That's the short answer.
Naama, thank you so much. It's just been great hearing from you. I know people are excited about this new test, and I know from the people who signed up and the questions we got, there's tremendous interest in your research as well. As I indicated, while these are very challenging times with the coronavirus, cancer's here to stay. And over 600,000 Americans will die this year, and many, many thousands of Israelis will die of cancer. And that will continue on into next year and the year after and the year after if we don't conduct research as a way of introducing better and more therapeutic alternatives. So I hope that all of you enjoyed the presentation today. On the screen are links to where you can donate so that we can continue to support the kind of outstanding research that you heard today. Dr. Maizels, pardon me?
Sorry. I wanted to say that I'm extremely grateful to ICRF. It's really, the funding was really crucial for us and helped us to advance our science on cancer. So,
well, it's very generous of you to say, and of course it means a lot to us. That's why we come to work every day. So Naama, thank you very much. Dr. Maizels, thank you. And to all of you who joined us, I'm very appreciative. Thank you very much.
Thank
and
you.
Goodbye.
Bye-bye.
Thank you. Goodbye.